Hello everyone, we are back and we have an exam coming up less than a week now on March the 26th. And I thought of doing something different today. Instead of just recording a regular lecture, continuing with the material regarding energy metabolism, diet, and weight control, what I decided to do was to go through a few practice questions. There's some of you who emailed me saying that you, in exam number 1, you studied well, you felt that you knew the material quite well, but when it came to actually taking the test, you didn't do so well as you expected. And some people ask me, oh, is there anything I can do to improve? What should I do? What is it that I may be doing wrong? So it's hard to say because I don't know each and every one of you. Each person is different when it comes to testing. But what I'm going to do today is just go through a few questions and try to show you the logic or the rationale when you actually look at a question and the important things that you need to pay attention, so you can actually use the knowledge that you have to your benefit. And it's also important to mention that if you only memorize information, it makes it really difficult for you to extrapolate or to analyze a question and then confront what the question is asking with the knowledge that you have. And then it makes it difficult for you to actually pick the right answer. So what I'm going to do is just show how we look at the options we have in each question, make decisions based on knowledge that we have, and then pick the question or the answer that is the one that is supposed to be picked for each question. So let's get into it and see how things go. But before we do this, I just want to remind you that on the 24th, Dr. Joss and we are holding a Q&A session where you're going to have the opportunity to ask us as many questions as you wish within that time allocated for the Q&A session. So today will be like a warm-up for questions and also for the exam, but be also aware that you can ask further additional questions or any clarification you have on the 24th when Dr. Joss and I are holding a Q&A session. So let's get to it. So the first question is, which statement below is correct? So first, when you're taking a test, make sure that you understand what the question is about. So here I'm asking the correct option below. So we have 5 of them. So we're going to start with A. So it's an 18, 2, delta 9, delta 12 is an omega-9 fatty acid. So this is actually looking at your knowledge of how the nomenclature and how you read a fatty acid, and you can tell if it's what kind of fatty acid it is. So when we say 18, 2, delta 9, delta 12 is because we are using the delta nomenclature. So we are counting double bonds from the carboxyl end on, and then we see that this fatty acid has 18 carbons, 2 double bonds. The first one from the carboxyl end starts at the carbon 9, and the second one is carbon 12. Now, how do I know if this is an omega-9 fatty acid? Well, we have to start counting the same fatty acid now from the methyl end. And if we start counting from the methyl end, what you really do here is you subtract 18 from 12. So it's going to give you an omega-6 instead of an omega-9. Okay, so you go from the methyl end and you start counting double bonds or carbons, and you see where the first double bond occurs, and you're going to see that the first double bond will be in the position 12. So if you subtract 12 from 18, you come up with 6. So it's an omega-6, not an omega-9 fatty acid. So that basically eliminates option A. So option B is all saturated fat are liquid at room temperature. Well, we know that saturated fat doesn't have double bonds, so which means that the melting point is high. So at room temperature, they are solid as opposed to being liquid at room temperature. So all saturated fat are liquid at room temperature. That's false. That's not true. So that also eliminates option B. So option C now, triglycerides may contain saturated fatty acids, MUFAs and PUFAs. Yeah, triglycerides, they are formed by one glycerol backbone, and you have 3 fatty acids esterified to that glycerol backbone. And those fatty acids can be saturated, monounsaturated, and polyunsaturated fatty acids. So that, and it depends on the diet. So if your diet is rich in all these different fatty acids, you're going to store in your adipose tissue triglycerides that also contain the same ratio of fatty acids, and those could be saturated, MUFAs, and PUFAs. So option C seems to be correct here. But before we close the deal here, let's move to D. So hydrogenated fats are always saturated. Well, not necessarily, because you can take a polyunsaturated fatty acid and you bombard with hydrogens. What happens is when you do this, it is called hydrogenation, you undo some of the double bonds, but you may still end up with 1 or 2 double bonds. And very often what happens is when you hydrogenate, those double bonds, they shift from being cis to being trans. Actually, that's the way how we make trans fat. So the food industry actually uses this process to make trans fat, but you have to have at least 1 double bond there. So hydrogenation does not always or necessarily convert a unsaturated fat in a saturated. It can convert in an unsaturated fat in the form of trans fat. So that also eliminates option D. And E, oleic acid, which is 18(1), is classified as a PUFA. Well, by definition, a polyunsaturated fatty acid has to have more than 1 double bond, so at least 2. So in this case, oleic acid is not classified as PUFA, it's classified as MUFA. So that's an easy one. So eliminates that option. So again, the correct answer here is C. I hope you understand, you got an idea of how we go one by one, and you actually analyze that option. Read well the option before you make a decision to make sure that you go all the way to the end, because sometimes the first few words of an option seems correct, and you say, oh, this is correct, and you just go and pick that option, and then turns out that in the end there was something, a couple words there that completely makes the question wrong or invalid. So make sure that you read from beginning to end every option, not only the title of the question, but also every option from beginning to end, so you know exactly what's going on. Number 2, which statement below is correct? Again, I'm asking you to pick the correct option that you have below. So essential fatty acids belong to the omega-3 family. So this is option A. Essential fatty acids belong to the omega-3 family. Well, we know that there are 2 essential fatty acids. This linoleic and linolenic. One is omega-3, the other one is omega-6. So therefore, we cannot say that they belong to the omega-3 family, because they belong to both omega-3 and omega-6 families. So that eliminates option A. Pepsin breaks down ester bonds in the stomach. So this is option B. So we know that pepsin is a protease. It breaks down not ester bonds, it breaks down peptide bonds, the ones that actually keep amino acids together and form a peptide. So those are called peptide bonds. But ester bonds, they're formed between, for instance, fatty acids and glycerol. So you have glycerol, you have 3 esterifications to 1 glycerol, and those form ester bonds. So pepsin breaks down ester bonds in the stomach? No, because pepsin breaks down proteins, and ester bonds are in lipids. So this is not correct. So that also eliminates option B. Trans fatty acids, option C now. Trans fatty acids makes fat less creamy. Well, we also learned that one of the reasons why the food industry adds trans fatty acids to food is because it reduces the chances of being oxidized. So it extends the half-life of a particular food on the shelf. So it's important from a commercial point of view, but also from the point of view of the user, the person who is going to actually eat that. It makes it also more creamy. In other words, you can look at it if you were like a fat. If you took a fat that was basically liquid at room temperature, it would be difficult for you to actually, like margarine, to actually spread on your bread. So now when you hydrogenate and you make it into trans fat, what happens is that it also raises the melting point. So now it becomes more creamy at room temperature. So option C is obviously not correct, because it says that makes less creamy when it actually makes it more, or creamier, if you will. LDL particles contain only ApoB48. No, it's not correct. LDL doesn't even have ApoB48. Remember, if you look at an LDL particle, and if you go to page, actually, in your book, if you look on page 38 of your book, you have like LDL. LDL has B100, which is the remaining apoprotein that stays there. It's not B48. B48 you find in chylomicrons, but LDL particles do not contain ApoB48. It's ApoB100, because it comes from VLDL that also contains B100. So that also makes that option invalid. And E, desaturases transform single ester bonds into double bonds. Well, by definition, this is what a desaturase does. It actually breaks down or converts a single bond into a double bond. Remember, the reason why we have specific essential fatty acids is because delta 9 and delta 12 desaturases humans don't have. So what happens is that you cannot actually add a double bond or convert a single bond into a double bond by specific desaturases. So by definition, desaturases transform single ester bonds into double bonds, which makes it the correct answer. All right. Moving on. Again, which statement below is correct? So humans do not have desaturases and cannot make PUFAs. Well, we just said that, yeah, humans don't have two desaturases that are important to make linoleic and linolenic fatty acids, which are considered essential. But what the option is telling you is that humans do not have desaturases and cannot make PUFAs. Well, humans can make PUFAs, and they do have desaturases. They don't have two specific ones that are required for linoleic and linolenic fatty acids to be made in our bodies. But all the others, there are other polyunsaturated fatty acids that don't have desaturations in those specific positions that you require for linoleic and linolenic that you can actually make. So humans do have desaturases and they can make PUFAs. They can't make only two of them, but that's because they lack specific desaturases, but that's not what the option is telling you. So that eliminates option A. So glycerol and fatty acids form chylomicrons glycerol and fatty acids from chylomicrons form triglycerides in the subcutaneous adipose tissue. Well, what's the problem here? We learned that glycerol, when chylomicrons deliver fatty acids to the adipose tissue, what happens is that fatty acids, lipoprotein lipase cleaves the triglyceride and releases fatty acids and then releases glycerol. But glycerol, we learned that is in chylomicrons. They don't enter the adipose tissue. They actually travel through the blood and go to the liver. So glycerol from chylomicrons is not used to esterify fatty acids and store triglycerides in the white adipose tissue. What happens is that, as you remember, we studied this. What the adipocytes do is they use sugar, glucose, to make their own glycerol, and then they esterify that. They use that glycerol that came from glucose to esterify fatty acids and make triacylglycerol. So glycerol and fatty acids, they are not both from chylomicrons to make triglycerides in the subcutaneous adipose tissue. The adipocyte, the fat cell, requires glucose to make its own glycerol, and then use that glycerol that came from glucose, not that came from the triglyceride that was broken down from chylomicrons. It uses the glycerol that came from glucose and then esterifies fatty acids. So fatty acids are true, they come from chylomicrons, but glycerol, no. Glycerol comes from glucose, and the glycerol that is released when the LPL breaks down triglycerides in the adipose tissue is released into the circulation, and that glycerol actually travels to the liver, and the liver uses it for gluconeogenesis to make glucose from glycerol, or you can also use that glycerol in the liver to esterify other fatty acids, because the liver can use glycerol, free glycerol, to make new triglycerides, but the adipose tissue cannot. So it requires glycerol that comes from glucose. So B is also incorrect. C, cholesterol undergoes beta oxidation in the liver. Well, we know that cholesterol cannot undergo beta oxidation. Fatty acids can undergo beta oxidation, and it's a very well established process inside of the cell, specifically then into the mitochondria. Cholesterol is not oxidized. Actually, cholesterol, the only way you eliminate cholesterol is when you produce bile and you release it in the feces, because it's really difficult for your body to break down, undo a cholesterol molecule. So again, it doesn't undergo beta oxidation. C is also incorrect. Phospholipids are amphiphilic molecules because they react with both water and lipids. Well, that sounds correct, because we learned that phospholipids, they are usually located in the membrane of cells, and they fit very well that purpose, because the cell membrane needs to be, or it establishes a barrier between the outside and the inside environment of a cell. So you need to have, because we have molecules that need to go in and out of the cells to be exchanged with cells, and some of those molecules, they are hydrophilic, and some of those molecules are hydrophobic. Some of them have affinity with water, and some of them have no affinity with water. So they are hydrophobic, which is the case of fat, for instance, as opposed to glucose, which is hydrophilic. So you have to be able, and because the membrane of a cell is the barrier that actually selectively allows molecules to move in and out, you need to have something that is located in the membrane, or molecules in the membrane that can actually communicate with both amphiphilic or with hydrophilic or hydrophobic molecules. So in this case, phospholipids, they have that particular feature. They are characterized by being able to react with both water and lipids, and therefore they are called amphiphilic molecules. So that makes D the correct answer, and obviously E, none of the above, is also incorrect. So here we go. D is the correct answer for this question. Next one, with regards to sterols. So which statement below is correct? So now it's specifically related to sterols. Remember, sterols are a class of molecules that have those sterol-like structures, or the ring, or that core structure that is typical of sterols. And if you go back, if we actually go back to the book and you look at the on page 30, we have here the typical structure of sterols. Yeah, they have this 4-ring core structure called cyclopentanoperhydrophenanthrin. So it's page 29 actually of your book. You see that we talk about sterols. So all the molecules that have that 4-ring core structure we call sterols. And obviously the most the one that we talk a lot is this one that is called which is cholesterol, that has 1, 2, 3, 4 ring structure right there. So here we say that the option says together with which statement below is correct with respect to whatever sterols are. So let's see what option A tells us. Together with phospholipids, they make the majority of animal fats. This is wrong, because what makes up the majority of animal fats is triglyceride. We have a small portion that is in the cholesterol form, but the majority of the lipids or the fats is not phospholipids and sterol, it's triglycerides. When you eat a piece of steak, when you eat any animal food that contains fat, the majority of the fat that you're getting from that is not phospholipids and it's not sterols. It's triglycerides. So that makes option A wrong. They're present in lipoproteins and cell membranes. We learned this. We know that cholesterol is a molecule that is cholesterol in the case of animal derived foods. They are present in membranes of cells along with phospholipids. So looks like B is correct. Now let's see what C also says. Are present in animal and plant derived foods. That's true, because cholesterol is in animals and ergosterol is in plant food. So both of them have their own types of sterols. So with respect to this broad category of sterols, then they're present in both animal and plant-derived foods. Now, if I were to specify, if it were with regards to cholesterol, then this would be wrong, because cholesterol you don't find in plants. It's only in animal foods. So, but because I'm not specifying if it's what kind of sterol I'm dealing with here, it's just general. Yes, it is true. They're present in animal and plant-derived foods. So we have B and C here that seem to be correct. Let's see D before we again close the deal. Cannot be synthesized endogenously. Well, that's not right. Again, because we know that there is a endogenous cholesterol synthesis pathway. So cholesterol is not an essential nutrient or lipid. And the reason for this is because we can actually make our own cholesterol. So endogenously means internally. We don't need to get it from the diet. You can actually make it yourself. So in every cell in your body, mostly liver, that is very good at this, can make lots of cholesterol. So we do have the ability to synthesize endogenous cholesterol. So cannot be synthesized is wrong. So that also eliminates option D. So we are with the option E, which says B and C, which is actually correct, because option B and option C are both correct. And there we go. This is the correct answer for E. The next one is, which lipids below derive from the action of lipase in the small intestine? So here what we are asking is, once you have lipase acting on lipids, what is the product of the action of lipase? And specifically in the small intestine, because you could have lipase in the mouth, you could have lipase in the stomach, but we are dealing here with lipase in the small intestine. So specifically, what the question is asking, what comes out of a lipid droplet once lipase act in the small intestine? So you can go, if you want to confirm what we're going to say here, you can go to page 32. But because before we go there, let's analyze one by one. Free cholesterol and lysophosphatidic acid. So lipase does not act on cholesterol or in your lysophosphatidic acid. What lipase actually does is breaks triglyceride. So cholesterol and lysophosphatidic acid are two things that are present in fat, but they are not, they don't require lipase to be available there. What you need to have is an emulsification of the fat, and that's what bio does. Bio emulsifies the fat, breaks down a big lipid droplet that contains cholesterol, lysophosphatidic acid, triglycerides, phospholipids in general. So every fat soluble molecule that is there will be emulsified by bio. Then what lipase does, so it opens up space so lipase can go and break down ester bonds. And those ester bonds, they are found in triglycerides. So free cholesterol and lysophosphatidic acid are not really dependent on the lipase in the small intestine. B, 2 megs and cholesterol. Again, 2 megs is correct because when you cleave a triacylglycerol molecule, you cleave one fatty acid from a tag, you make a DAG, and then you can cleave another one and you can make a MAG. So it could be 2 meg because it's a fatty acid that remains esterified to glycerol position 2. So that would be a 2 meg. But cholesterol is not. So you have partially, that's why I say always read the question from beginning to end and make sure that you pay attention to the detail, because 2 megs is correct, but cholesterol is not. So that also eliminates option B. C, long chain fatty acids. Yes, because you cleave one fatty acid from a triglyceride, for instance. Now that fatty acid is free, and we call long chain fatty acid, or abbreviations LCFA, or long chain fatty acids. Correct. So we also have 1 megs because you have glycerol esterified with only one fatty acid at position carbon 1 of the glycerol, and you also have 2 megs, the ones that you take off position 1 and 3, and you only leave a fatty acid in the middle of the glycerol. So that's the position 2, so it's a 2 meg. And that's what seems to be correct, because actually if you go to page 32, we have a diagram where we have the triglycerides, so the TG, big TG droplet, and you see here the pancreatic lipase. What it does is it breaks it down and releases long chain fatty acids, 2 megs and 1 meg. And that now can, along with all the other like lysophosphatidic acid and cholesterol, because of the emulsifying effect of bio, they all form micelles, and those micelles can now actually enter the enterocyte and be converted inside of the enterocyte into a chylomicron. So again, C is correct here. Before we pick that option, let's look at D, cholesterol and 1 megs. So it's similar to B. So the difference is that I switched the position. Instead of saying 2 megs, instead of saying meg and cholesterol, I said cholesterol and meg. So the difference is that B has 2 megs and D has 1 meg. But what makes this question invalid, or this option invalid, not a question, the option invalid, is that I'm also including cholesterol. And cholesterol does not depend on the activity of lipase in the small intestine. And then E, all of the above. Obviously not, because we know that there are at least 3 options here that are not correct. So the only correct option here is C. Now, the next one is, which below are features of HMG-CoA reductase? Okay, I'm asking here, what are the characteristics, or what are features or things that are actually particularly true for HMG-CoA reductase? Remember, HMG-CoA reductase is the main enzyme, or the rate-limiting enzyme in the process of endogenous cholesterol production or synthesis. Again, you can go to page 30, where we actually have the entire pathway that goes from fatty acids to cholesterol. And right in the middle here, we have what we call HMG-CoA reductase, which is the rate limiting step of this process. So this is what I'm trying to, or I'm going to ask here is what options below are actually features of HMG-CoA, so we can actually attribute to HMG-CoA reductase as one of its functions. So A, regulate intracellular cholesterol content. Well, remember, HMG-CoA reductase is more active or less active depending on the intracellular cholesterol content. So if you don't eat any cholesterol, not at all, zero cholesterol from the diet, and your body needs cholesterol because it's a very important molecule for many different functions in your body. Now the cell, if the cell doesn't get it from the diet, the cell says, well, if the cholesterol levels inside of the cell is low, then the cell makes its own cholesterol, so activates the pathway, the endogenous cholesterol synthesis pathway, and it does so by activating HMG-CoA reductase, so which means that the enzyme does regulate intracellular cholesterol content. So if you have low cholesterol content, it will increase it. If you have too much, it will shut down its activity to reduce the cholesterol content. So it works as a regulatory step or a regulatory mechanism of how much cholesterol is inside of the cells. B, its activity is regulated by intracellular cholesterol. Yes, remember, when cholesterol is produced, what it does is it has a negative feedback mechanism that it goes, as it accumulates inside of the cell, it goes back and inhibits the activity of HMG-CoA reductase. So it does regulate, so its activity is regulated by intracellular cholesterol levels. It's highly active in the liver. Yeah, we also learned that because actually the liver is the organ that probably produces the highest amount of cholesterol in your body, and mostly because the liver produces a significant amount of bile, and bile is essentially made up of cholesterol. So liver has to have a continuous supply of cholesterol. So if liver doesn't get enough cholesterol from the circulation or from the dietary sources, it will make quite a bit. And it's, again, the HMG-CoA reductase is very active in the liver. So you can find it basically in page 30 again of your book. Is allosterically inhibited by cholesterol? Yes. What do we mean by allosterically inhibited? So remember that I'm saying the same thing that I was saying for instance, in option B, except that I'm using now the specific mechanism by which the enzyme activity is regulated, which is allosteric. So cholesterol does inhibit HMG-CoA reductase allosterically, meaning that it acts away in a site, or it regulates the activity of an enzyme in a way that is distant by interacting with a site that is distant from its active site of where the catalytic activity of the enzyme takes place. Instead of being in a specific pocket where the catalytic action of HMG-CoA reductase acts or exerts its effect, cholesterol actually interacts with some other areas far away from that catalytic site, and then changes the conformation of the molecule, and now the molecule cannot, or the enzyme cannot actually do its job if you are inhibiting it, so which is the case here. So with that, pretty much all four options you have here, even some of them are very interrelated, or I would say that there is some repetition in terms of what they say, but they all, in different ways, they are saying the same thing. They are actually playing around with the idea of what the enzyme actually does. And all the four options that you have, A, B, C, and D, are correct. So obviously, the correct answer is all of the above. Let's do one more. Choose the correct statement below. Sorry, I didn't highlight this one. I didn't underline. But again, as I said in the exam, I'm trying to always underline what the important word of this question is. So I'm asking you to pick the correct answer below. So now this is dealing with, again, PCSK9 increases, option A, PCSK9 increases LDL uptake by the liver. Well, what do we know by PCSK9? We know that once it's released into the circulation, it actually binds to the LDL receptor, and if the whole complex, PCSK9 and the LDL receptor, internalizes. So which means that the LDL receptor now disappears from the surface of the cell. So if it disappears from the surface of the cell, there is less possibility of LDL actually binding to the LDL receptor and being uptaken by the liver. So which means that PCSK9 does the opposite of what the option is saying. PCSK9 reduces LDL uptake by the liver. It does not increase, because it actually promotes the degradation of the LDL receptor, so you have fewer LDL receptors sitting on the membrane of liver cell, or the hepatocyte, so there are less opportunity for LDL particles that are in the circulation to actually be uptaken by the liver. So option A is the opposite, what it should be, so it's wrong. Lack of PCSK9, option B now, lack of PCSK9 causes hypercholesterolemia. Well, it's the abundance of PCSK9 that causes hypercholesterolemia, not the lack of PCSK9, because PCSK9 inhibits or impairs the ability of liver to clear LDL particles, and LDL particles, we know they are rich in cholesterol. So now if you don't have PCSK9, what you're going to do is you're going to allow more of the LDL receptors to take up LDL particles and then remove cholesterol from the circulation. So actually, instead of causing hypercholesterolemia, you should actually lead to hypocholesterolemia. It's actually, again, the opposite. So what is important here is that you understand what PCSK9 does, and then now you interpret the question based on the knowledge that you have. But you have to think about this. You have to really put your brain to work here. You have to process the question, and then that is that thought process that really allows you to pick the right answer. See, that's exactly what I'm doing. I'm just using the knowledge that we have and apply to the question, and obviously look at each option and see if that makes sense or not. So option A and B, they are actually telling us the opposite of what PCSK9 does. So they're wrong. So option C, inability to produce ApoC2 causes hypertriglyceridemia. Remember now, what do we need to see here? What's the function of ApoC2? So remember, chylomicrons and VLDL, they contain ApoC2, both of them. And both chylomicrons and VLDLs, they are rich in triglycerides, which means they're carrying triglycerides to peripheral tissues. So if it's a chylomicron, it's because it's coming from the diet. If it's a VLDL, it's because it's coming from the liver. So it's endogenous production of triglycerides. But in both cases, they are going to have to deliver their cargo, their triglyceride content, to peripheral tissues. And one of the things that is crucial for this to occur is that on the surface of a chylomicron or a VLDL, you have to have ApoC2. ApoC2 is the apolipoprotein that actually works as a flag and also like a kind of like a receptor to bind, to anchor chylomicron and VLDLs to the LPL enzyme that is on the surface of the capillary. So once that binds to it and the interaction of ApoC2 with the LPL, it actually not only anchors the particle momentarily, but it also activates LPL. LPL is lipoprotein lipase. And what that does is that it cleaves triglycerides and then allows fatty acids to go into the cells, so the cells can actually take and use those fatty acids that are being delivered by these two lipoproteins. But it's essential that you have to have ApoC2, because if you don't have ApoC2, the cells become blind to chylomicrons and VLDL. They cannot see it because it is the ApoC2 that is hanging on the surface of the particle that will allow it to anchor and actually interact with the cell, with the capillary level, and then allow LPL to actually exert its effect, which is cleave the triglyceride and allow fatty acids inside of the cell. So if you now look at the option, inability to produce ApoC2 causes hypertriglyceridemia. Well, it makes total sense, because now if you don't have ApoC2, let's say you have a genetic disease or an inborn defect, genetic defect, that you cannot produce ApoC2. So if that's the case, now you cannot even put ApoC2 on a chylomicron or VLDL. So what happens is that all those chylomicrons and VLDLs that are produced, they remain in the circulation. So and because they have lots of triglyceride in them, it develops hypertriglyceridemia. So again, option C is correct. So it makes sense. Again, you have to take the knowledge of what ApoC2 does and apply it. Again, it's not only memorization. You need to be able to process the information. You need to be able to understand what you're reading. So don't just memorize. Think about things. Think about how you can apply the knowledge and make sense of it. And this is a typical question that is requiring you. It's a higher level, and I acknowledge that. It's a higher level of difficulty, because what I'm doing here is I'm forcing you to think. It's not just take a knowledge that you memorize. You need to be able to think and process the information and be able to make sense of it. So it's application. It's a question, typical question of application. So option C so far is correct. D, LPL and ApoB48 are produced in the liver. Well, we know that ApoB is right, but LPL is not produced in the liver. LPL is lipoprotein lipase. LPL is produced by the cells that actually take over, or that cleave, the cells that actually use take fatty acids from the particles. And so in order for them to be able to extract fatty acids, because those fatty acids, they are in a triglyceride form, they are esterified to glycerol. So you need to be able to separate them, disassemble the molecule to take the fatty acids. So you need LPL. So LPL is produced by the cell where that fatty acid will actually be used. So if you look at adipose tissue, it produces its own LPL. Muscle produces its own LPL. Liver produces its own LPL. So every cell in your body that can use those fatty acids are coming from chylomicrons or VLDL, for instance. They have to produce their own LPL, lipoprotein lipase. So LPL and ApoB48 are produced by the liver, and so ApoB48, yes, but LPL, well, you could make the case here, well, Roland, this is not totally, it could be confusing here, because LPL could also be produced by the liver cell itself, but keep in mind the liver can uptake all the lipids, because it can take in all the lipids, not because of LPL breaking it down, but because you have an internalization of particles via receptor-mediated process. So D, again, is not the correct answer. So none of the above is not right, because obviously you already said that if you don't produce ApoC2, you're going to have hypertriglyceridemia, and we said that is the correct answer. So C is the correct answer here. I think this question is probably a higher level of difficulty, and I acknowledge that, but that's on purpose, because I want you to be able to think about things, to be able to interpret a question and not just use simple memorization, because not a few questions are based on simple memorization, but majority of them you have to think. Okay, let's do one more and then we'll stop here. I think this is an easy one. And all I'm asking here is, which lipoprotein sequence, or which sequence of lipoproteins below contains the highest to lowest phospholipid content? So I want to know phospholipid now, and I want to see which one has the highest to the lowest. So what is the sequence of distribution of phospholipids in these different lipoproteins? So option A. So option A gives it starts. So it's saying that chylomicron is higher than VLDL and VLDL is higher than LDL. Well, we know that chylomicrons, they're not rich in phospholipids. They're rich in triglycerides. So chylomicron and VLDL, they are rich in triglycerides. So LDL actually has more phospholipids than the other two. So actually it's not a valid option here. It's invalid. Now, HDL, chylomicron, and LDL. So HDL, we know that is the highest, the protein or the lipoprotein that has the highest content of phospholipids. So we know that anything that starts with HDL here will be a good possibility here. So now what we need to see is HDL higher than chylomicron? Yes, but is chylomicron higher than LDL? No, then that's not the right answer. Is LDL higher than HDL? No, because HDL is higher than LDL. So C is also not correct. Now let's look at D. HDL, VLDL, LDL, and chylomicron. So it's starting with HDL, it goes to VLDL, it goes to LDL, and then to chylomicron. So, but again, we have a problem here because we have VLDL ahead of LDL. And then we have the last option, which is HDL, LDL, and chylomicrons. So the correct answer is E, because HDL is higher than LDL, and LDL has a higher content of phospholipids than chylomicrons. And again, you can go to your, there is another diagram here, page let me see what page is this is. Yeah, here. It's page 36. Page 36 gives you the composition of all the different components, and you can see the phospholipids is higher in HDL than which is the orange color here. So it's higher in HDL, and then the second highest LDL, then is VLDL, and the one that has the least amount of phospholipids is chylomicron. So if we go in the order from highest to lowest, obviously the only option is HDL versus HDL, LDL, and last chylomicron. The only option that fulfills that requirement is option E. So you don't need to memorize the numbers. All you need to know is have an idea of what the distribution of these different lipids is in these different lipoproteins. I don't know the numbers. If you ask me the numbers, I don't know, and I don't memorize them, but I know proportionally which is the main component of each of the lipoproteins. And it's true for phospholipids, but we could be asking what is the opposite. Like, what if I asked what about cholesterol? What if I asked about what is the lowest to highest triglyceride content? So there are different variations of the same kind of question that all you need to know is the distribution of these different lipid contents, lipid forms within the lipoprotein. So I think that gives you an idea of this is not the same level of knowledge that it's required. This is, I would say, an easier question because you can, just by having a good idea of what the composition of the lipoproteins is, you can pretty much answer the question. And you can eliminate several of them just by looking at what the highest would be. I mean, if you start with chylomicrons, knowing that it's the lowest, that option alone is invalid. So we have only three possibilities here. That are two of them we eliminate, A and C, and then we have three possibilities that we have to look with a little bit more detail, and then you have to know a little bit more than what the AGL, or you have to know a little bit more about the distribution of lipids in the other lipoproteins, and that will do the trick for that. So with this, I think we are going to stop today, and I hope this is of any help to you, because again, the most important thing when you take a test is that know the stuff, know what the subject is about. But when you study, don't just memorize, because if you do, first of all, you're not going to be able to interpret questions that are more application instead of memorization. Second, you'll probably forget it. You won't remember it, because once you finish this, it's gone. You memorized, and then if you don't really apply it, you end up forgetting it. So if you understand the knowledge, it becomes useful, not because of this particular test or this particular course, but you're going to be able to use this knowledge in other areas of your life and also in other courses you take, because there is overlap of information that you can actually use and you can actually interact and can help in putting together all the information that you get when you get a degree in kinesiology. So it goes beyond the idea of an exam. So thanks for watching. I'll probably see you on Wednesday, on the 24th, prior to the exam, and we'll be able to address other questions in a Q&A session. Thanks again for watching, and bye for now.